Hello everyone, my name is Sofya, and today I will talk about genetic monogamy and mate choice in titty monkeys. This is a part of my PhD that I did at the German Primate Center in Göttingen. Genetic monogamy is a rare phenomenon, and this is well illustrated by an example of birds, where historically most of birds were considered socially monogamous or pair-living. But with the development of molecular genetic methods, it became clear that actually most of birds have extra pair paternity, including even the white stork, the traditional symbol of monogamy. And in mammals, genetic monogamy is even more rare than it is in birds, and so far, strict genetic monogamy with 0% of extra pair paternity has been found only in 6 mammal species. And several other species can be considered mostly genetically monogamous with very low proportion of extra pair paternity. One of the reasons genetic monogamy is so rare is that pair-living animals can have extremely constrained mate choice. First, because mates become unavailable once they are paired, and second, because animals might face a conflict between having to choose for direct benefits, such as, for example, good territory, and indirect benefits such as genetic qualities of a partner. And to compensate for these constraints, animals might engage in extra pair copulations. In addition to deal with one particular risk of constrained mate choice, the increased risk of inbreeding, animals might disperse because dispersal would disrupt close kin associations. But we still don't really understand whether dispersal has to be sex-biased to generate enough genetic dissimilarity between males and females to prevent inbreeding. And also, there is still a lot we don't know about dispersal in pair-living mammals, because in some species both sexes seem to disperse similar distances, but in other species females seem to disperse further. And now I move to the object of my study, the coppery titty monkey. Titty monkeys are very good candidates for being genetically monogamous because they have several traits that are associated with low levels of extra pair paternity in mammals. First, they are pair-living, with one group consisting only of one breeding pair and the immatures. Second, they have long-term pair bonds between males and females, and third, they have biparental care of infants. So to collect my data, I went to Peruvian Amazon, and there I collected fecal samples from 14 family groups of titty monkeys. Then I genotyped all my study animals on 18 microsatellite loci using the genotyping by sequencing approach. And I also sequenced a fragment of mitochondrial DNA, and I used behavioral observations because in addition to the genetic part of my project, my PhD also had a behavioral part. So, to see whether titty monkeys are genetically monogamous, I did parentage analysis using microsatellite and mitochondrial DNA haplotypes and also direct observations. And I will go directly to the results. I found no cases of extra pair paternity in my study population. And here on this map, I show the animals for which I reconstructed parent-offspring relationship with the different colors corresponding to different mitochondrial DNA haplotypes. And my analysis showed that all the social fathers in all 17 cases of assigned paternities were identified as genetic fathers of the group offspring for 9 groups. Although, of course, with the sample size of only 17 assigned paternities, we cannot exclude that there is some level of extra pair paternity in this study population. Nevertheless, this makes the coppery titty monkeys one of the very few mammal species where no extra pair paternity has been found. Next, to see whether titty monkeys avoid inbreeding, I analyzed mate choice. And here I used microsatellite genotypes to compare relatedness between mates in real and randomly generated mating pairs sampled from the breeding pool. And here I found no evidence of relatedness-based mate choice because there was no difference in relatedness in real and randomly generated mating pairs. But what was interesting was that despite no evidence of relatedness-based mate choice, the average relatedness between pair mates was still very low. And here on this map I show the pair mates of each of my study groups, again with different colors corresponding to different mitochondrial DNA haplotypes. And we can see that only in one pair, in group 6, the pair mates are related on the level of second-degree kin. And also what we can see here is that no mates share mitochondrial DNA haplotype. Next, to see whether this low relatedness between pair mates could be explained by dispersal, I analyzed the genetic consequences of dispersal. And here I wanted to see whether dispersal distances differ between sexes. And to answer this question, I compared relatedness and mitochondrial DNA diversity between sexes. And here the idea is that members of less philopatric sex would be less genetically similar than members of more philopatric sex. And I also conducted special autocorrelation analysis to see whether animals preferably disperse fixed or varying distances. And here I found that both sexes disperse similar and varying distances because I found no differences in relatedness or mitochondrial DNA diversity between sexes, and I found no special genetic structure in either sex. So, to summarize my results, I showed that my study population of copper-eating monkeys is genetically monogamous, and I found no evidence of active inbreeding avoidance through mate choice, but despite this, the average relatedness between pair mates was still very low. And this suggests that even non-sex biased dispersal, as long as it is not constrained, can be sufficient to prevent inbreeding. And here I should say that the results on mate choice and dispersal should be treated as preliminary because the sample size was quite small, and also the special scale of the study was quite limited. And in the future I plan to check if these results still hold in higher density populations, for example in fragmented populations. And if you want to find more details on this study, it is all published in this paper. And with this, I would like to say thank you to my supervisors from the German Primate Center, to all my field assistants, and to all of you for listening. Thank you.